My talk is on control of protease activity in S. pombe using gene silencing approach. As far as the introduction, thousands of genes they have been identified and cloned using recombinant DNA technology, and their expression and production of interest, they are important both for basic research and practical applications. Hence, the demand for foreign gene expression system is increasing rapidly. Many kinds of foreign expression gene expression systems have been developed using different cells like E. coli, streptomyces, etc., yeast, plant cells, most eukaryotic and prokaryotic. Each system has their own merits and demerits, and we have to choose depending upon our needs. Prokaryotic cells, they are very commonly used as a host, as their manipulation is very easy and handling is easy, production cost is low, everything is low, like Sir was telling, he has chosen to produce an E. coli because the production cost is low. But when we are going to express high eukaryotic gene, sometimes it results in an inactive product without proper folding and post-translation modifications. This is the basic drawback we face when we try to produce any high eukaryotic protein in a prokaryote. Then they can be solved using different techniques like if we produce higher proteins in high eukaryotic animals only, like plants and cells, but manipulations are not easy, and handling plants and animals, and again they are not cost effective, the basic thing. So yeast, it comes as a good host for eukaryotic protein productions. Being a eukaryote, this is a eukaryote. Molecular biology research is well developed, genetic manipulation is easy. It has been used in fermentation history for a long time, so we know the culturing methods and various techniques. And it is almost free from endotoxins, so it is safe to use. Amongst yeast, fission yeast S. pombe is an attractive host model for high level production, as it shares many molecular, genetic and biochemical features, and it is distinguishable from other yeast through its ability to proliferate by fission rather than budding. And many of cellular processes, they are like mRNA splicing, post-translation modification, they are closely related to high eukaryotes. So, and moreover, it has a developed Golgi apparatus and galactosyl transferase that is not found in other yeasts, and that is required for proper production, proper folding and secretion of high eukaryotic proteins. So it comes as an ideal host. But the major hurdle in effective production and purification of proteins in S. pombe is it produces a number of proteolytic enzymes which degrade the foreign foreign protein, whichever is produced, whichever we try to produce in it. It becomes very significant if we wish to secrete the recombinant protein. It is secreted out, it secretes a number of degrading proteases, they cut the protein, and the production is very low. So a number of attempts have been, scientists are working, they have tried a number of methods, like we can control the cultivation conditions or medium contents, we can change addition of protease inhibitors, but again all have their own limitations. Every methods they have, like if we control growth conditions, then and adding protease inhibitors, they might also inhibit the growth of the cell also. Then cultivation time, it cannot be shortened, because for high cell density batch cultures we need high production, we need both cultivation time large. Then genetic manipulation, it comes as a potent method to control this. Gigahama et al, they have tried this, and they have a good number of publications and published work. What they did, they disrupted the S. pombe protease gene, which is which is acting upon the gene of interest which we are going to produce, and then they designed specialized vector for producing different higher eukaryotic proteins. Here in our study, we tried gene silencing approach for reducing the activity of proteases using RNA interference by siRNAs. This is RNA interference is a process of post-transcriptional gene silencing in which double-stranded small interfering RNAs, they cause sequence-specific degradation of mRNA sequences. And it is basically a two-phase process. Initiation is small regulatory RNAs, they are generated via processing of a longer double-stranded RNA precursors by RNAs. Here you can see in this, sorry. This is the gene from which a double-stranded RNA is generated by protein AGO1. Then dicer acts on it, generates small interfering RNAs which go and bind to the RIST complex, and this is activated. It goes and cuts the target protein, target mRNA, and that is silenced. And it is not translated further, it is transcribed, but after transcription it's silenced by such RNAs. Major classes are siRNAs, microRNAs, and small interfering RNAs. Both miRNA and siRNAs, they silence at post-transcription level. So it was evident from literature that S. pombe has all the machinery which is required for RNA interference process. So it can be used as a model system to express siRNAs against proteases which are cutting the protein, desired protein which we are producing. So keeping all this thing in mind, this study was aimed to control the protease activity, by generating small interfering RNAs to silence that protease which is acting upon the model protein. So I used S. pombe as this expression vector PREP1, and this was given by East Genetic Resource Center of Japan, and these are the E. coli. It was used for preservation and expression of the vectors. First of all, what we did, we designed DNA construct of, first of all we chose a model protein, and I selected human parathyroid hormone as it's a very small protein and number of proteases acting upon it were small. So we chosen a protein, then it was designed using different bioinformatic techniques first, and this this construct was designed. I downloaded this structure amino acid sequence and a signal peptide sequence of carboxypeptidase which is of S. pombe origin. It was downloaded and it was applied 5' to this sequence so that this protein is secreted out of S. pombe, and then it was reverse transcribed using pombe codon usage so that it is properly expressed in terms of proteins, amino acids. Then this designed gene construct, it was commercially synthesized from JNAT, and it was applied in this vector, and it was used to transform E. coli DH5 alpha to preserve, and from where I then took it out. Then both this cloning of HPTH gene construct into REP1 plasmid, the plasmid which was used, both these plasmid vectors, they were digested with the enzymes, cutting enzymes ND1 and SMO1. They were digested, and you can see the products here, and they were eluted from the gel, and from then they were ligated, and after ligation I got this thing. This is the PREP1, and this foreign gene construct, it was added here after NMT1 promoter, and it was having CPY1, this leader sequence. Then it was confirmed using PCR technology that whether the protein is being processed. This lane shows the protein which was in the GNAT company, and this one from Rep1, which was added, in which it was added, and then protein expression was checked. A clear band of approximately 6.5 kD was found in SPV1, this pombe transformed cells. Then this protein was 160 mg per liter. The production secretory, the amount which was secreted was very less, and this was probably due to in the proteases they were acting upon it, and it was reducing its amount. Then to control that activity, we designed a DNA construct for silencing the YPS1 proteases, which was resulting from this MIROPS database. I tried to find out that which proteases were acting upon HPTH, and here it was found out to be this A01.056, which is YPS1 protein of S. pombe, and this is an aspartic proteases of Yapsin family. Then to control the proteases degradation of human parathyroid hormone by this gene silencing construct was designed against YPS1. This 653 base pairs open reading frame, it was cloned as an inverted repeat with the same sense and antisense arms of repeat separated by 53 bases of spacer containing the first intron of red 9. The intron was included because intron containing hairpin induces PTGS in plants with nearby 100% efficiency. So it was designed to make a hairpin-like structure. It was used here, the construct used here when spliced is presumed to leave a loop of 14 unpaired nucleotides and double-stranded RNA hairpin, if we can see it here. It is a structure like this, leaving this hairpin loop. This hairpin loop present of double-stranded structure with hairpin loop causes AGO1 to go, and this loop, and a double-stranded structure is formed, which is acted upon by dicer and produces siRNA. So this structure was constructed. And siRNA so generated will guide to degradation of target mRNAs via base pairing with complementary sequences. The design construct again was synthesized from JNAT, this company, and this time I added it into Rep2 vector of that same expression system, and the products, they were then purified again. It was cut, the vector was cut, and the design, this was added. This was the gene of interest, and they were then again eluted from the gel, ligated, and this is what I got in P-Rep2 vector system. It also has an MT1 promoter. There it is the YPS hairpin structure gene was incorporated here. Then this was the YPS1 was added, Rep1 was added to same SPV1 protein, this culture having YPS SPV, Rep1 this vector already. Then total RNA was isolated from both SPV and SP2 and were quantified from OD obtained as this, and they were found out to be good in a good concentration. Then mRNA level of YPS1 and SPV1, both the cultures, was determined by analyzing the relative gene expression system RT-PCR, and it was amplified by RealPlex2, and it was quantified with this 2 delta-delta CT method. The fold change in expression of YPS gene normalized to internal control was analyzed using this CT values provided from, not showing exactly. These are the CT values, and this is delta 2 delta CT, and this is 2 raised to power delta CT values, which shows here for YPS1 control it was 1. It is not coming actually, I put this from other slides, picked up. It is 1 here, and it was reduced to 0.3. And this graph shows the 2 delta DCT value of YPS. This is the YPS1 control, and this is the test in which siRNA, this gene silencing cassette, was added, and it showed 68% decrease in production of YPS1 expression, that is the protease. And here this gel shows this lane 1 as the YPS2, and this is YPS1. This is the standard protein. And when we check the growth curves for both the SPV1 without gene silencing cassette and SPV2 with gene silencing cassette, it showed almost no change in growth pattern. Hence, then comparison of HPTH expression system was done. Proteins were isolated from both the systems, and they were in the gel you can see from SPV2 with gene silencing there was increase in this production of protein, and then HPLC was done, and from here you can see there was almost the increase was like area 0.16 microgram per ml to the increased to 0.95, almost 9-10 times increase, 8-fold increase in concentration of HPTH, the foreign protein. So this ultimate goal of the study was to develop an pombe host strain for effective production of proteases with the sensitive heterologous proteins by reducing the effect of selective proteases which are acting on the heterologous system. Here the particular protease is knocked down instead of completely knocked out. Its expression is lowered, so the actual function of the protein in the cell is not disturbed, and there is no effect on growth of the cells, but there was effect on its silencing properties, and the production of model protein was increased. So here we succeeded to develop a custom designed protease deficient pombe strain for effective production of HPTH by reducing the effect of selective proteases on which was acting on HPTH protein. That's it. I acknowledge University Grants Commission for giving a grant, and thankful to Dr. Praveen Balbalkir for her valuable guidance, and again UGC for providing me the travel grant for attending this conference. Thank you.
We have just maybe a question. One question. Yes.
Thank you. My question is, after you express the protein into the yeast, is it possible to do some screening whether the protein can improve the growth of the yeast, like a drug screening or osmolarity screening?
Pardon?
You express the protein into the yeast.
Yeah.
Is it possible to do some screening after expression of the protein into the yeast?
Yeah, it is possible, but my aim was just to check the proteases activity to reduce that. So we didn't go for that part, but if we need to use that protein for therapeutical purposes or other purposes, we would have to go for all that screening. We need to go through all the screening processes. But we didn't do because my aim was little different. And if we wish to use that, then we actually need to go through those processes also.
Okay, I think the time is almost up. So